People have known about high-altitude headaches since the time of the Chen Dynasty around 2,000 years ago, yet we still don't know what causes it except for low oxygen. My research is seeking to find a potential cause of acute mountain sickness by looking at its relationship to the patent foramen ovale and systemic inflammation. Acute mountain sickness is a debilitating condition that occurs when individuals go too high in altitude too quickly. The incidence of acute mountain sickness in literature is variable, but the main idea is that the higher and faster you go, the more likely you are to get it. Acute mountain sickness is associated with hypoxemia, which is low oxygen in the blood, and anything that makes hypoxemia worse contributes to acute mountain sickness. And one thing that can make it worse is a hole in the heart. The patent foramen ovale is a hole between the right and left atria of the heart that allows deoxygenated blood to enter the left atrium directly. It is present in around a third of the population, and overall this decreases the amount of oxygen in the blood and contributes to hypoxemia, which can lead to an increased incidence of acute mountain sickness. Studies have shown that subjects with a patent foramen ovale may be more susceptible to developing acute mountain sickness. We looked at some of the reasons for why this might be, and specifically we looked into biomarkers of inflammation. Studies have shown that there are increases in inflammation when we go to altitude, and a few studies have shown an increase in inflammation in subjects who develop acute mountain sickness compared to subjects who do not. Preliminary work from our lab has found that inflammation decreases after getting the patent foramen ovale closed. It was hypothesized that the patent foramen ovale subjects would have a higher level of systemic inflammation at baseline compared to subjects without the patent foramen ovale, and upon exposure to simulated altitude of 15,600 feet, inflammation will increase in both groups, but it would be highest in the patent foramen ovale group, which would increase susceptibility to acute mountain sickness. To do this, we tested 34 men and women with and without a patent foramen ovale. Subjects entered the environmental evidence chamber with conditions simulating 15,600 feet, with oxygen at 11.5%. While in the chamber, plasma samples were taken and acute mountain sickness scores were collected using the gold standard Lake Louise scoring system. A 13-plex human inflammation panel was used to measure the concentration of 13 inflammatory markers in the sample. We found that 83% of subjects with a patent foramen ovale, but only 61% of subjects without a patent foramen ovale got acute mountain sickness. Figure 1 shows that from baseline to 10 hours of high-altitude exposure, there was a general increase in inflammation. Figure 2 shows that in general, subjects who did not get acute mountain sickness had larger changes in inflammation than those with acute mountain sickness. Figure 3 shows that subjects who did not have a patent foramen ovale had greater changes in inflammation and appear to be protected from getting acute mountain sickness. Figure 4 shows a significant difference in inflammatory marker interferon alpha-2 between the subjects with and without the patent foramen ovale at the 10-hour time point. Figure 4 also shows a significant difference in inflammatory marker interleukin 12p70 between those with and without acute mountain sickness at the 10-hour time point. In conclusion, we found that there is an increase in inflammation with exposure to hypoxia, and generally the increase in inflammation was beneficial, which is contradictory to our original hypothesis. We found that only interferon alpha-2 was associated with the patent foramen ovale, which suggests that it may be involved in increasing the susceptibility to acute mountain sickness. I would like to thank the Department of Defense for funding this project, as well as Caitlin DeMarco and Andy Levering for all of their help with this project. Thank you.